Okay, the lecture today on Wolbachia. Let's see. Okay, so I study Wolbachia. Wolbachia is gram-negative. It's a bacteria. It's obligate, intracellular. This is the first obligate intracellular we've talked about. So what does that mean? Yeah, I couldn't quite hear that.
You can only live within the cells.
Yeah,
It's taken
so-
out of the body.
Very good. So this is a unique bacteria that can only live inside of eukaryotic cells. So here's a eukaryotic cell. Some obligate intracellulars are a little different. Some will live like in vacuoles. Others will live directly in the cytoplasm. Wolbachia seems to live essentially like in like a little vacuole that it surrounds itself with, in contrast to maybe something like Rickettsia, which lives in the cytoplasm. Okay. So it can't live outside. That means it also has fairly reduced genomes. So Wolbachia genomes are about 1,200 genes in size, and it's actually also about 1.2 million base pairs in terms of genome size. So that's a pretty small genome size for bacteria. So we study Wolbachia because it has some really interesting phenotypes. So it can manipulate insect reproduction. So it lives inside of a lot of different things. It lives inside of nematodes. It lives inside of insects. It lives inside of crustaceans, mites, and spiders. So there's lots of kind of insect-like things, nematode-like things that have Wolbachia inside them. And in the insects and in the mites and spiders, it causes what we call RP, which is reproductive parasitism. So every time people talk about Wolbachia, there's different like instances of it, and there's different strains of it, and they have different phenotypes. So if you had like a sliding scale of parasite to what would be I'm blanking on the word, like mutualist. So mutualistic, like they're helping the host, or parasitic, where it's hurting the host. There's different Wolbachias all throughout this like sliding scale. Okay, so there's some Wolbachias that live in bed bugs that are thought to provide like B vitamins. So the bed bug eats blood, and the blood is kind of a nutrient deficient source, and so it's thought that the Wolbachia in the bed bug provides some like nutritional benefits. So it's more mutualist in that sense. And then there's other cases where it's more parasitic. Some of these are actual instances of reproductive parasitism where the only reason it's there is because it can manipulate the reproduction of the host to like maintain itself. So we'll talk about some of those phenotypes. So some of the different reproductive phenotypes are list: male killing, feminization, parthenogenesis, and CI. And there's actually like a few others, and they get more complicated. But in general, these different phenotypes are all reproductive manipulations, and they all serve one purpose, which is to create a situation where there's more females. And the reason that, why do you think that the reason that Wolbachia wants there to be more females is because it's another characteristic of it is that it's maternally transmitted. I don't know if it's 2 T's, 1 T. Maternally transmitted. Okay, so if you had a male and a female, let's say fruit flies, and let's say they're infected with Wolbachia. In the male, the Wolbachia live or concentrate in the testes, and in the females, they concentrate in the ovaries. There are some somatic infections of Wolbachia, so if you like sample from the brain, you'll find periodically some Wolbachia. If you sample from different body parts, you'll find periodically some Wolbachia. But for the most part, like the vast majority of the bacteria are condensed in the reproductive tissues. And in the males, there's a little bit of caveat in that in the males, it's not in adult sperm. So it lives in the tissue that produces sperm, but it's not actually in the adult sperm, and so it's not sexually transmitted. So if like an infected male mates with an uninfected female, it will not pass the Wolbachia infection. The only way that Wolbachia can move to the next organism is through maternal transmission, which is through the eggs. So in this, it is in mature eggs. And every time the female lays an egg, it's densely saturated with Wolbachia. And in part, that's because the nurse cells are, remember when we drew all those diagrams, there's these nurse cells that surround the egg, and they're also heavily infected with Wolbachia. And so every time the female lays an egg, all the nurse cells dump their infection into the egg as the egg is getting laid. Okay, so it's perpetually maternally transmitted. It's not paternally transmitted, okay? And that's the main, I guess, reason why it has evolved to manipulate reproduction in a way that creates more females, because it likes to have more females because every female is a new host where it can pass on thousands of its brothers and sisters, brother and sister Wolbachia, to the next generation, okay? So let's discuss these phenotypes a little bit more. Okay, so male killing is, let's say you had the egg fertilized by the sperm, okay? And let's say you have another egg fertilized by the sperm. Let's say these eggs are both laid, both freshly laid by a mother that is infected with Wolbachia, okay? In male killing, what the Wolbachia can somehow sense is they can sense, this mechanism is not quite known yet, but they can sense the genotype of the egg and sperm mixture. And they're able to tell and discriminate whether it's male or female. And in the case of male killing, it will figure out, okay, this one's a male, and it will kill that egg. And the reason that this can be selected for is because this male, so in like evolutionary thinking, the reason that this can be selected is because this male is a dead end. If this were to grow up, the Wolbachia in there are stuck. Whereas if this female grows up, these Wolbachia can transition to the next generation. And so if there's a situation where these brothers and sisters might actually be competing for resources, if they're competing, that can create a situation where the Wolbachia essentially learn how to kill the males, and then this one is more likely to survive, because if there's a limited amount of nutrients in the wild, you'd rather have more females. So this can alter sex ratios. So in some insect sex ratios are 50-50, and other times they're not, and this is a situation where Wolbachia might tweak those ratios to favor more females, okay? So that's an example of male killing. Feminization. Feminization is essentially when, if you have a sperm, let's say you do another thing, sperm, egg, let's say this one is genetic male, this one is genetic female. So in some insects, so again, like some of these phenotypes are only in specific insects. Some of them are specific to a certain genetic context. But in some insects, the sex can be manipulated, okay? And so feminization is when the Wolbachia, if they're in a male, they sense the genetics, the genetic context of that, and they're able to convert it into a female, so that as it develops into a female, and then that becomes a new outlet for reproduction, passing on the Wolbachia. That's feminization. Parthenogenesis, this mechanism is a little bit different, and there's a little bit There's subtle like variations on parthenogenesis, and it gets very complicated depending on the organism. But in general, if you have eggs, let's say you had an egg that's fertilized and an egg that's not fertilized. In certain insects, unfertilized eggs might develop as a certain sex. So there's certain situations where an unfertilized egg might develop as a male, or there's certain situations where an unfertilized egg might develop as a female, depending on the insect. Okay, so things can get complicated. But Wolbachia can cause a situation where, let's say you had a fertilized egg that was going to develop into a male. It can essentially destroy the set of chromosomes that come from the father, and then it reconverts back to a situation where it looks like it's unfertilized, and that can lead towards a path where it develops as a female. Again, this depends on the wasp, it depends on the context, but there are certain situations where Wolbachia can select or cause, through genetic mechanisms, the genotype to actually, the sex of the organism to actually change. Okay, 4, CI. That's what I actually study. We'll do a whole thing on CI. Okay, so here's CI. CI stands for cytoplasmic incompatibility. Okay, so I will put this on the test. This is a Punnett square. Let's do females, and let's do males. Say these are fruit flies, and let's say this one is infected, and this one is infected. So this is plus Wolbachia female. Okay, so here's the deal with cytoplasmic incompatibility. If an uninfected male mates with an uninfected female, that's just a normal sperm in an uninfected egg. The outcome here is survival. This is just like a normal insect, and there's no Wolbachia. No Wolbachia passed on. So this would maybe like, in some insects, wild type means actually natively infected, but you could consider this just kind of like a wild type cross with no infection. Okay, in this case, where you have an uninfected male mating with an infected female, you have a sperm fertilizing an egg that is infected, okay? These survive, and they inherit Wolbachia, because the female is infected, okay? Here is what's called the CI cross. So this is where you actually get cytoplasmic incompatibility. So in this cross, the Wolbachia that live in the male's testes, they produce some kind of modification. That's what I study, is I study the genetic mechanisms of how this modification sort of gets imprinted onto the sperm, okay? And it essentially is a conditional sterility where it can sterilize that sperm, okay? So in this case, where that sperm with some modification that came from the Wolbachia is fertilizing an egg that does not have Wolbachia, in this case, the outcome is death, because that sperm is sterile. And obviously, if it's death, there's then no Wolbachia passed on to the next generation, and the female is not infected. Okay, in this cross, the final cross, this is what's called a rescue. This is the rescue of the CI phenotype. So if this male who had Wolbachia, which sterilized his sperm from some genetic modification, if that sperm combines with an egg that has Wolbachia, okay, these Wolbachia have evolved to know that that sperm is sterile, and they're able to fix it. Okay, so they're able to remove whatever the modification was to make it functional again, and these embryos survive, and they inherit Wolbachia. Okay, so if I ask like what is CI, you should be able to kind of like draw this. And the main one you want to remember is especially this one, because that's what causes the, this is the CI cross, and then this is the reciprocal rescue. Okay, so let's think about what happens at what this causes to happen at a population level. Okay, so ask yourself, which female would you rather be? Would you rather be this female who's infected or this female that's not infected?
Infected.
Yes, why?
Because all of her offspring will survive.
Yes, so in a mixed population where you have some individuals that are infected or uninfected and some individuals that are infected, if there's a population that looks like that, you would rather be her, because it doesn't matter if you mate with him or him, all your offspring always survive. And as long as there's no detriment to having Wolbachia, as long as there's no sort of like fitness reduction to having Wolbachia, you would much rather be her, okay? Because all your offspring will survive. Whereas if you're her, depending on who you mate with, and usually with Wolbachia, there's no mate discrimination. They cannot tell, they cannot sense whether or not an insect has an infection. They can't tell. So it'd be a huge disadvantage to be her, because if you accidentally mate with him, all your offspring die. Okay, so if you were to graph from the population perspective, time on the x-axis, or generations, and percent infection on the y-axis, okay, you will see Wolbachia infections go like this. So they will increase until they hit equilibrium frequency, like a fixation rate, okay? And that fixation rate, depending on the insect, maybe it's something like 90 to 100%, okay? So usually it's not 100%. There are some insects like Culex pipiens, which is a mosquito, that almost is 100%. But it's usually not absolutely 100% for a few different reasons. So one reason is that maternal transmission is not 100% efficient. So if you have one female and she lays 100 eggs, if it was a 100% maternal transmission, all 100 would be infected. That's not usually the efficiency. Usually the efficiency is something like 90 to 95%. So there's usually like 5% of embryos that come out not having Wolbachia. That's what causes this to reach quite 100%. That's one. Another thing is that there are some minor like fitness costs to having Wolbachia. So it's like you're carrying like an extra load, and that can also contribute, so fitness cost can also contribute to sort of like reducing this from reaching 100%. Okay, but in general, like the pattern is that if you have a mixed population of insects and you have CI, you will see Wolbachia increase in the population until it reaches like a high infection frequency. Does that make sense? Okay, so there are two main uses for this in biotech, probably more, but right now 2 uses in the field. So uses 1, 2. What do you think are the 2 uses, given they all revolve around CI? One essentially uses this, and the other essentially uses this. Okay, so one of these is sterile insect technique, and the other is gene drive. Okay, so for sterile insect technique, this process can be utilized as the mechanism to produce sterility, okay? So all you need to do is remember in sterile insect technique, we talked about how you would build like a factory, and in that factory you would rear a bunch of insects. So the way the sterile insect technique works with Wolbachia is you go out in the wild and you collect, let's say you collect a mosquito, and you want to use sterile insect technique with that mosquito. You can create an infected line with Wolbachia. How would you do that? Yes, you'd microinject it. So this is what we're doing with certain insects, is you can do this two ways. One is called a cytoplasmic transfer. Another one would be a direct sort of like purification of Wolbachia. Okay? So let's say you had a deep mel egg. Drosophila melanogaster is natively infected with Wolbachia. And let's say you collected a mosquito egg, which was from that blue mosquito. A cytoplasmic transfer is when you poke this with an embryo, or you poke this with a needle, you suck up a little Wolbachia, and then you take that needle and then you immediately spit those contents out into the mosquito egg. That's the cytoplasmic transfer, and you're transferring Wolbachia. That can transfer Wolbachia into the mosquito. Okay, so now you have infected mosquitoes, and those infected mosquitoes are different from the wild population. So if this is wild, and this is now the plus Wolbachia blue mosquito, you can rear these in the factory, okay? And then as long as you sex separate them, you'll take all the males, and then when you do this cross, this one to this one, that will produce cytoplasmic incompatibility because it's an infected male, and those Wolbachia will modify that sperm and combine with an egg that's not infected. So that gives you CI, that gives you death. And in general, this is a simple idea of how sterile insect technique would work with Wolbachia. In practice, it's a little bit more complicated because in practice, the mosquitoes that they do this with are typically Aedes albopictus. Aedes albopictus mosquitoes already have two Wolbachia, so they have W alb A, W alb B. So you have, I'm telling you that there's a mosquito that they do a sterile insect technique with. It's already got, let's say, a green Wolbachia, and then let's say a black Wolbachia. It's already got two Wolbachias. So how can you create CI if the wild females already have Wolbachia in them? First, let's understand why this would be a problem. So imagine you grow these mosquitoes in here. Remember, this is our wild. And it's got black and green. If you grow this and then you mate this to that, these Wolbachia, that's going to create a sperm situation that looks like this, where it's going to have like double modifications. It's going to have a black modification and a green modification, and it's going to be in an egg in the wild, from the wild population that has green Wolbachia and black Wolbachia. So these Wolbachia will be able to recognize those not the mutation but the modification, and then they'll be able to fix that. So this would actually produce rescue. So how do you actually get CI?
You add a red Wolbachia?
Yeah, you got to add a new Wolbachia. So what they did, this is in China, they added a W pip, which is from a different mosquito, Culex pipiens. So they made a triple infected mosquito, which they grow, and then now they do this cross, and now the one that they can't fix is the red modification. So what this shows you a lot of different things. One, the application is a little bit more complicated, but it still works. And the general idea is that you just take an insect that doesn't have Wolbachia, or maybe it doesn't have the right combination. You infect it with a different combination that creates sterile sperm, and then you can use it for a sterile insect technique. Lost my train of thought. Oh, the other thing that this implies is at the molecular level, this actually implies some really interesting features. It implies that the way that CI works, the way that the mechanism works, is some kind of like specificity, where there's some kind of like red modifier. Imagine like there's some kind of red modification, and then there's some kind of like red rescue factor, and these things match. And there's also some kind of green modification with some green rescue factor, and then there's some black modification with some black rescue factor. And this only matches this, and this only matches this. and this only matches this. And so you can't like have a situation where the red one is rescuing the green modification. So what does this suggest actually at like a genetic level, the molecular level, how this might work?
It's like receptor.
Yeah, that's exactly what I thought. I mean, I wouldn't say it is a receptor, but you're getting to exactly the right thought, which is it's probably a protein-protein interaction that's highly specified, where there's evolution of whatever this thing is, there's a direct evolution where this thing is specifically evolving to recognize this in a very tight manner. And whenever this changes, this also has to change. And so it's very much like a receptor kind of situation where there's this, and sometimes we call this a lock and key. It's a lock and key situation where the biochemistry has evolved, where the proteins are actually evolving specificities, and then they're evolving specific recognitions. That's the best hypothesis for how this works. And I study the genes of how that works. Okay, gene drive. So that's how you would use this for sterile insect technique. Let's talk about the other one, gene drive. So, okay, I'm figuring out how best to introduce this. This gets kind of complicated. Okay, so there's some other interesting phenotypes with Wolbachia. If you have a mosquito and you infect that mosquito with a new, what we would call heterologous Wolbachia. What does heterologous mean? Where did that word also come up this semester? It comes up in the context of recombinant protein expression.
Like chimerics?
I wouldn't say chimeric. Heterologous usually means you're taking something, you're putting it in a different context or a different setting. So sometimes you say heterologous protein expression, where you're taking like a protein from some organism, putting it in a different organism, expressing it. So heterologous Wolbachia means you're taking a Wolbachia from somewhere else and you're putting it in a new context. So in the case of the gene drive, what they did is they take W-mel. So also for more context, when you're talking about Wolbachia strains, they usually listed with a W and then the species of the host. So this would be Wolbachia from what organism? Yeah, Drosophila melanogaster. So if you take Wolbachia from Drosophila melanogaster, and this is now Aedes aegypti, so this is not Aedes albopictus, different mosquito, Aedes aegypti, which wild type is not infected with anything. If you take Drosophila Wolbachia and you infect them in Aedes aegypti, this mechanism is still not quite understood. We don't quite know how this happens, but when you do this, it stimulates the insect immune systems of this mosquito, and it makes it less likely that it would spread flaviviruses. So the actual phenotype of a heterologous Wolbachia infection in Aedes aegypti is that it lowers vector competence. That's a long word for saying that it reduces the transmission of flaviviruses. So flaviviruses are things like dengue, Zika, yellow fever, West Nile virus, all those nasty diseases, those are flaviviruses. And if you have a heterologous Wolbachia in Aedes aegypti, it lowers the vector competence. So this is actually kind of like a good mosquito. You would rather be in a world where these mosquitoes populated your environment rather than a world where you had normal Aedes aegypti. So the concept now is if we could create this mosquito and then spread it and get it to replace the wild type population, which is not infected, you would much rather have this, okay? And so you can use the CI as a gene drive system, because remember I showed you that graph where if you have infection percent, it goes up like this, okay? So it increases. This is what this is. That is a gene drive. So a gene drive is a situation where you're taking some in theory, some genotype, although like this maybe is not genotype per se, it's an infected mosquito, but you're taking some thing that you want and you're driving that into a population and it's replacing the wild type population, okay? So you can use CI, the rescue feature of CI, which produces this effect to drive this phenotype into a population of mosquitoes. And this is what they did in Australia, where in Australia, in certain parts of Australia, the native Aedes aegypti used to look like this, and it no longer looks like that. Now it looks like this. And there have been no local transmissions of dengue in Australia since they did that. So it's a way to replace and change insect populations to produce insects that have more favorable phenotypes. So these are two usage cases of Wolbachia. Are there any questions about that? How much time do I have left? Oh, you're gonna ask?
I was just gonna ask, have they seen stuff like that drive evolution of the virus itself? Like, is it finding different?
Oh, that's an interesting question. Like, would then the virus needs to respond in order to get spread? Oh, that's an interesting question. I don't think that they've seen that yet. How would the virus evolve in response to that? I have not seen that in the literature. Good question, though. This is like cutting edge. Okay, how much time do we have left?
10 minutes.
10 minutes. Okay, so let's talk about the mechanism of CI. Okay, so CI mechanism. That's what I study. And there are two genes that are associated with the CI phenotype. Why does it make sense that there are like only two genes? In theory, there could be many genes or one gene. Why does it make sense inherently, logically, intuitively, that there should be two genes?
Because it's probably on the plasmid?
No. Well, what would that have to do with the plasmid?
You don't fit so much on the plasmid.
It's actually not on the plasmid. It's on the chromosome. So somewhere on the chromosome of Wolbachia, there's these two genes.
Ploidy.
What?
Ploidy.
No.
Two genes.
No, okay, think about the phenotype. Remember, think about like the Punnett square, where you have the induction of CI and you have the rescue of CI.
Does it need like the lock and key mechanism?
Yeah, that was my hypothesis. Again, like this is still actively studied, and this is still like we fight about this in the field if you're really nerdy. But to me, it inherently makes sense that two genes would be associated with this phenotype because one gene is probably causing induction. We call this the toxin. And another gene is causing the rescue.
Oh, wait, is this a toxic antidote system?
Yes. So this is a toxin antidote system. Although again, like we fight about that. Like that's my hypothesis. All my career has focused on studying this. Okay, or not all my career, but much of my career has focused on studying this. So there's two genes. We view one as a toxin that induces CI. One is an antidote that rescues CI. Okay. How would you prove this hypothesis using synthetic biology? How could you prove this hypothesis?
Knockout rescue.
So that's a good idea. That would be the best experiment, is if you could do a knockout in Wolbachia, you could just knock out toxin gene, and in theory, you would then have no CI. The problem is nobody can do genetics with Wolbachia. So nobody has ever successfully done a targeted knockout, and nobody has ever successfully done a targeted mutation. So that is off the table in some sense. Although it's still a good idea that you could also pursue. How else could you also pursue that idea?
Like a specific protease?
Well, you could look at genetic diversity. So remember, there's many different strains of Wolbachia that live in many different insects. Most of them cause CI. And so you could sequence a bunch of genomes, and you could find a strain of Wolbachia. You could find a strain. With no CI, and then you would look at the operon. Okay, so they did do this. There's a strain called WAU, and in WAU there's no CI, and there's also no rescue. And when you look at those genes, they are completely gone. Okay, so in that genome, there's none of these genes, and you get none of the phenotypes. There's also a strain called WMA, which has rescue, but no induction of CI, okay? When you look at the WMA genome, it has this gene, But the second gene is pseudogenized, which means it's got a mutation, it's broken. So this is the genetic phenotype, or the genotype that causes this phenotype. And then there is there another one? I'm not remembering. I mean, these are kind of like the big ones. So essentially what I was telling you is you can't do knockouts, but if you could find natural knockouts, which they did, you can look at the genetic context, and that genetic context supports this hypothesis. What else could you do to actually like prove it? That's more correlative. What would be direct proof? How could you prove it using synthetic biology? Think about all the tools I taught you. How would you do an experiment or build something that could test whether or not these genes cause CI? What organism would you want to work with?
Drosophila.
You could work with Drosophila, okay? Good idea. We did do that. How would you work with Drosophila? What would you build? And what techniques would you use that I've taught?
What do you mean by prove it?
Prove that this causes CI and that this rescues it. How would you prove that?
Can it be knocked out in Drosophila?
Well, so if it's natively in Drosophila, that means that the ovaries are infected with these bacteria that have their own genomes, and you can't knock it out in that setting. But we've talked a lot about Drosophila like genetics. So what could you do? I don't want to give you the answer. I mean, I feel like this is worth.
Could you get a sample of Drosophila PCR or some piece of its DNA and look for those, and then have just a huge number of Drosophila mate and see which ones have the gene, which ones don't, which ones get CI, which don't?
So you're real close. But instead of sort of like sampling Drosophila or PCRing, like we want to actually like insert a gene.
Can it build a plasmid?
Yes. Okay. So we could build. Okay. So what plasmid would you build?
One with the toxin and the antidosis.
But you're not going to give a plasmid to an organism.
Okay.
Like you're right that we would first need to build a plasmid, but the intention is not to build a plasmid. The intention is to actually do what?
Would you use a nurse cell promoter like an ovary?
So now we're actually starting to get very detailed. Okay. So let's think about this plasmid. Okay. So yes, let's build a plasmid. Let's put toxin, and let's put a germline promoter. We could build this plasmid. What would we want to do with the plasmid? If we're going to test it in our system, which is going to be Drosophila, how are we going to deliver this and test it?
Put it in the egg.
We're going to microinject it, yes. Okay, but what does it need to get inserted? I'm trying to like review from like how you actually build stuff in Drosophila. What kind of system are we going to need? So first, recognize a problem that you can't give a plasmid to all the somatic cells of an organism. So you could give a plasmid to, in theory, a embryo, but that plasmid will not replicate in all those cells. So instead of giving it a plasmid, we give it a plasmid to do what?
Before blastoderm? It's like the.
Like there's one key word that
It jumps into the.
yes. Okay. That's exactly what I'm looking for. Like the point is you can't give it a plasmid. You have to give it a plasmid that's organized to integrate onto the genome. So you could either shoot this in with a transposon. That would be one way. Or if you wanted to do it in site-specific way, you would put the ATT. I can't remember if it's B or P. Let's say.
It's B because it recognizes the bacteria site.
Okay, there we go. So you'd have the ATTB site. If you did the integration properly, there'd be a 5C31 integrase in that embryo. okay? And there'd be a landing site somewhere on the chromosome for ATTP. This would then recombine onto the chromosome. Now we would have a transgenic Drosophila. So we did that. We would have a transgenic Drosophila whose genotype is now toxin. We can make it homozygous with that germline promoter. You take a male, and then what would be the actual experiment?
Made it with a wild type female.
You'd cross it to a wild type female with no rescue factor. And if everything was perfect, you should get 0 hatch rate. That would prove that this causes CI. This has been done, this experiment has been done for a strain of Wolbachia called W0. They took the toxin from W0, they inserted it onto a Drosophila chromosome, they did this cross, and they didn't get 0, but they lowered the hatch rate. So now you're starting to see like genetics and experiments are a little bit more complicated. I could tell you a ton of experiments we've done sort of like on this topic, but then the next experiment would be what? So if you could prove then that this caused CI, what would then be the next experiment?
Cross it with a female with rescue.
Yeah, make another female, make a transgenic female with the antidote, or the antidote with a germline promoter. They did this in the same type of organism. So they took the corresponding antidote from W0, Wolbachia no. They put it in a transgenic female. They did this cross, and then that raised the hatch rate to give you rescue. We also did this for, so essentially, so this data, that experiment has been done for 2, what I would call orthologs. So imagine running out of space, running out of time. It's an interesting question of like, have you proven it? So imagine you have 10 Wolbachias. 10 Wolbachias. Okay, every Wolbachia has one of these operons, and they're unique. And that experiment and that data has been done for 2 of these, and it's been done for other ones, and in other ones, it's a little bit more complicated. So in some cases it's working, some cases it's not. So it's like, have you proven it? I don't know. But you understand the complexity?
Yes.
Yeah. Any questions? How much time do we have left?
We're done.
We're done. Okay. Anyway, that's Wolbachia.